Good afternoon. I'm going to talk about very more detailed bioinformatics aspects and their translational angle. So even if you don't have this much background, you try to understand why something very basic knowledge of protein understanding is very important. Whatever research you are doing, either in plant science, in animal science, human physiology, or in daily routine activity, if you don't understand the main reason, the cause, which is 90% cases is protein, and how these protein formed, what are their amino acid constitutions, why these amino acids are so important, and they are behaves differently. So that actually the aim of this talk, so that you have at least knowledge. So you can see here the topic of the talk is computational microscopy, need of the hour. So in experiment you already heard this word microscopy. So why I used this term computational microscopy, because I am going to funnel down and narrow down you at the level where you can talk, you can see the information about atoms. Because if you understand atom, that atom constitute altogether to make amino acids, then amino acids combine together to make peptide, then peptide to polypeptide, and polypeptide to protein, and then protein 1D to 2D, two-dimensional to three-dimensional, and then three-dimensional to four-dimensional. And then active form of the protein, inactive form of the protein, how these proteins changes when you are in disease state, when these proteins are behave differently when you are in normal rather than in disease condition, whether it is a normal flu or a pathetic lethal disease like cancer or Alzheimer. So let's start the talk. This is the overview of my research area, where I'm mainly interested in the development of the four components. So which hit means here the small molecule, the drug which you are seeing in the market. The initial stage of your drug is called hit, which is have all the properties of to become a drug-like candidate, but initially in the discovery phase it called hit. So hit discovery in different methodological section, then mechanistic studies, automated pipeline generation for to resolve this complex problem, and then peptidomimetics, which is the most advanced stage of computational driven small molecule designing. So for these four purposes, the system which I am using is protein ligand, protein DNA, protein peptide, and protein protein interactions. So the theme of this understanding in more general, the core of the interest is molecular recognition. So you all heard about it. What is actually the molecular recognition? So at the basic level, when you any process, when signal communicate from one state to another state, if you feel hungry for anything, you get a signal. So how this signal communicate from one chamber to another chamber in your body, in from cell to organ, from tissues in the distribution process. How this communication happen? This communication happen through only one process, and that is molecular recognition. When one molecule means when a partner, like here is the ligand, interact with this protein. This is a transporter protein which absorb your glucose. So when glucose enter from here and it transport, it have some particular signal which communicate when it interact with the another protein. Similar way, the transcription and translation process, there the protein interact with the DNA. So this is basically the molecular recognition process. If you understand this molecular recognition process at molecular level, at atomic level, then it would sound better to talk about drug discovery or drug identification. So when we obtained these potent hits, we used molecular computational strategy. We talked to with medicinal chemistry and biological assays in vitro to design potent lead molecule which have druggable properties. So basically, why my core area of interest is protein, because it belongs to 90% cases in disease and disorders. Protein shape is important, is a blueprint for all the biological functions. So the biological functions going on in any cases, even this center or northeast is rich with the plant activities, but that activities is also governed by these proteins only, the nucleic acids only. So it doesn't matter whether you apply your bioinformatics knowledge, your drug discovery ideas in plant science, in animal science, or in medical background. You have to understand these criteria where only one thing is exist, either nucleic acid or amino acids. And if you understand the nucleic acid and amino acids at molecular level, you can design your own program, you can design your activity, you understand your research work very detailed. So these actually, the proteins are not a static structure. If you see in the book, textbook or others, these proteins are written there, but they are not a static structure. They are dynamical in shape, and the dynamic properties is the intrinsic behavior of the protein. So if you target one protein for your understanding, it would not give you exact response, exact knowledge where you can understand the protein dynamics in detail. So how protein conformation change in dynamics, internal motion, and when protein interact with the other proteins, any protein-protein interaction. So how it change in normal and disease state. These and one another important aspect, since protein involved in the cell signaling and communication, so it never works alone and it interact with the other protein. For example, if protein 1 recognize protein 2 for a particular function, for a particular biological function, for example, transcription, so the same protein 1 can interact with another protein, for example, third, to perform another function. So the same protein, how they decide and become a manager to decide, okay, now for this purpose, A will interact with B, for this purpose, A will interact with E, and A will interact with Z. So the same protein, how this communication happen, and if you understand this process in atomic level, you can plan to your work, and you can understand the result part in more detailed way. So the best example to understand the protein structure is crystallography. So crystals tells you how the protein formed, how this structures formed basically. But even though if you have a particular crystal structure for your problem, that's not going to tell you all the details about the protein dynamics. So do we need any advanced technology, computational method to resolve the limitations of crystal structure? Yes, we need it. So here I'm going to talk about two main things for the drug discovery point of view. First thing is develop binding site specific protein chemistry. How, if you know about your binding pocket, your architecture of your binding site, where you imagine your drug can go and bind. So from that area, if it is a static protein, that site is called orthosteric or allosteric site, where the drug bind and do the either modulate the biological function or perturb the biological function. Other is the interface site, which localized at the protein-protein interaction surfaces, and that is called interface sites. So to resolve these problems, I encompasses multiple techniques which range from rare event analysis, quantum calculation, molecular mechanics, molecular dynamic simulation, docking. So I pipelined all these tools to understand the protein dynamics for only one purpose, that is hit identification. So I'm going to tell you one successful story which funded by DST Serve project. I got this project, and I started from 2009 onwards, and we have at least 2 patents on it and a substantial publication in this area. So the story is about RNA-dependent RNA polymerase. I think you all know at least transcription and translation. So in transcription translation, one protein is polymerase is very common. So this polymerase belongs to bovine viral diarrhea virus. When I joined postdoc with the group in Germany, so they are working on this to understand the dynamics of this bovine. Bovine is very important for them because they depend, like we depend, our staple food is rice, wheat, but they depend on the cow and cow meat also. So for them, it's very important to investigate why this virus is so lethal for cows and cattle to destroy completely. If this infection happen, the cattle sure short it die in few months or few days, depend on the severity of the infection. So they all are interested to know and want to investigate design a small potent molecule. So we have a team of chemists, biologists, and computational scientists who design a molecule taking this crystal structure as a target. And from this target, we designed a molecule which is active enough, that is 19 micromolar. This is not going to do anything because 19 macromolar having all the properties of the drug-like property, but the team is wanted to develop more potent molecule, and for that we used more computational data. what we did, we started our simulation, started molecular dynamic simulation. So what is molecular dynamic simulation is actually, it starts from the crystal, taking as a starting point, as a seed value, then from Newton equation of motion, you apply the acceleration potential on that, and you generate multiple snapshots with dynamics, with motion, with water, with solvent, with concentration. So you mimic your system exactly similar as it is in natural behavior, and then you generate data which have a multiple snapshots. So what you get from the multiple snapshots, we find that rest portion is very static. There is no such changes during simulation. But this linker portion is very flexible. And this linker is, suppose when we go through in literature, we found that linker is important for escorting the RNA template towards the catalytic site. So we got the clue that if our molecule is 19 micromolar is okay, good enough, but it's not able to block this linker movement completely. So we have to take into account this area to design something, to add something in this molecule so that this linker can freeze on here and RNA template can block completely. So what we did, we take the amino acids from this zone and we generated the pharmacophore QSAR model to design this molecule. And this molecule block this property and it become 2 nanomolar concentration, and the potency is 2 nanomolar, which is till now the best potent molecule reported till date. So idea is protein structure is important. When you understand this protein structure, since protein is not a static molecule, you should understand how it moves along with the time. And if you understand this, then next question comes into the picture. If your molecule is working, how it is working? So the process is inhibition. If it is inhibiting your protein, how it inhibiting? So we not only developed a selective molecule for bovine viral diarrhea virus, that molecule is active on hepatitis C virus also, but at least 100-fold down. So it's a broad spectrum molecule which having the potency to block 2 viruses together. And when we investigate the possible mechanism of inhibition, which is this direction, so you can see these are the linker area which I shown you in the last image, which is in blue color. Now here is red. So this is the crystal property. If you see the black one is the crystal one, this is the transient position, and this zone we obtained during MD simulation. So you can see this space. This space is nothing but when RNA template enters into the binding pocket to do the polymerization. So the RNA template enters from this area, and that area is enough to do the polymerization in the active state of the protein. What happens when the molecule bind, which is in van der Waals state of this compound, this is the 2,2,7-G drug, when it enters, it squeezes all the arms of the zone. See, it interacts with this arm, it interacts with this loop, and there it completely blocks the tunnel. What happens? RNA is not able to enter. When RNA is absent, there is no polymerization. So we propose that this could be a possibility that when RNA template are not able to enter into the pocket, the RNA polymerization will stop, and because of this reason, maybe 2,2,7-G is more potent. But this was the totally done in computation, so we have the validation. So another collaborator from Spain, they performed a crystallography data, and when they developed, they found the same result. Means what we proposed from MD simulation, that this molecule bind at the template entrance site, interacting with the residues, doing a conformational change, not allowed RNA template to enter into the pocket, that is the mechanism of inhibition, turns completely true from the experiment that this molecule bind at the surface. So we confirmed, we published this work. So that's the way, means computational tools are strong enough if you have proper logic and arrangement, then you can understand and validate your results. Means dry lab can be possible to have advanced work than wet lab. You design your work so that you can prove in wet lab. The second, another interesting phenomenon is resistance. So we all know about that if a person is infected with HIV, one of the problem is the emergence of resistance. If a person is affected with cancer, cancer is also suffering with the emergence of resistance. So why this resistance is so important? So we also face the same problem with the same protein, that the molecule which is reported by many other authors and many people, they find a common resistant mutant in the same zone. And when we designed our molecule, we also find a common molecule in the same area. So it means that this zone is somehow related with the biological function of this protein, which allow this protein to render itself in a way so that it can kick out the effect of drug and makes itself so prominent so that it can perform the biological function. So why any protein produce the mutant? The intention of the protein is to generate mutant is to retain their biological function. And how they can do this retention of the biological function? By introducing a mutation which will be causing least stability state, means the protein cannot going to change, but it can throw out the drug effect. And that's we try to speculate the possible mechanism of resistance. How we did? We performed a longer MD simulation in multiple states, when there is no drug, when there is no substrate, when there is a drug but with mutant. And what we found, when in the normal state, when drug is there, they bind with the molecule with all the possible loops here, and there is no space for the RNA entrance site. So here you can see the RNA double-stranded RNA can easily enter, but when drug bind, there is no space. But when mutant occur, even though drug is present, but RNA template can enter. So what happened? The virus is smart enough. He introduced a small mutation nearby the zone where even drug is present or absent, doesn't matter. It make the space by which it can allow the RNA to enter, so that it can do the polymerization process. So we understand through molecular dynamics simulation, we did computational studies how this happened, and which is not only through computational. Then we later on we calculated and we performed the surface binding plasmon resonance reactions, and from experimental we prove that yes, there is no current. We did the efflux pump analysis and with the current, and we found that there is no current to flow here because the RNA can easily to enter in this site. So experimentally we confirmed that what proposed is true. So another interesting aspect of protein is the drug and protein binding interaction to calculate the thermodynamics. So I was seeing the syllabus, and anywhere most of the cases, this thermodynamic aspect, the concept of the thermodynamics is very important in terms of the stability of the protein when it forms the complex. So how this thermodynamics pattern change when it is in wild state and when it is in complex state. So this thermodynamic state, when drug binds there, it interacts with certain residue. To make the interaction pattern, we calculate the delta G or experimental IC or EC50 value. When drug bind in presence of mutant, which is COM-M, that is complex but with mutant, then the delta G varies. So, but protein don't want to vary so much. It want to retain their properties. So they did slight changes. So that's the reason sometimes association energy, which is calculated by Monte Carlo and MMPBSM techniques, are not able to reproduce the same data which actually reflect in experiments. So to generate the more accurate way, more robust way by which we can calculate, we can generate data which can able to reproduce the same thing what experimentally we are getting, we perform dissociation also. So in dissociation, what we are doing, we are trying to pull out the drug from the binding pocket. So if, as just assume, if this is the binding pocket and you did the association process, your drug is coming and bind to the pocket, it will generate a delta G. It's a thermodynamics process. But when you do the reverse, when you try to apply the force to pull out your small molecule from the pocket, it is a dissociation process. And by doing dissociation, when it interact with the other residues, it will take that much energy. So from association dissociation, when we perform these two experiments, then we can comment that what actually the delta G is. So we performed these two experiments to confirm that not only in rotavirus, not only in dengue, this RNA template entrance site is common in most of the RNA strand viruses, negative strand RNA viruses, and this molecule which we develop are able to have effect since it's occupy the template entrance site. So the take-home message is you cannot rely only on the crystallographic data. You need to understand the active and inactive state of your protein dynamics. What does it mean? Active and inactive state means on and off state of your protein. And these on and off state provide you enough atomic level information for your small molecule designing. Second thing, the association process, which is calculated by MMPBSA, is not always the gold standard for in silico drug efficacy measurement. You have to perform other experiments like dissociation when you deal with the flexibility of the protein. And in that scenario, the dissociation association ratio will provide the net delta G. Okay, so this was the first story. When you have your static structure and you want to develop your lead molecule, what process you have to do and to get a small molecule which is potent enough. The another interesting story I'm going to talk about is the protein-protein interaction interface. So why this protein-protein interaction is important? Apart from static structure, this PPI is more complex and challenging. Why? Because there is no any detailed defined architecture of the binding pocket exists in interface site. And since it involved in most of the biological process and disease, anything happen in the body is through a cell signaling process, and cell signaling is nothing but the PPI interaction. So if you want to design a molecule from the interface of the protein-protein interaction, your pocket should be very defined and clear. But unfortunately, this pocket is more and more challenging and not able to track your residue-wise quantification to design some molecule because of there is no any thumb rule to design for the small molecule. So we have to learn from the different success stories from which is already reported. And the success story means how nature performed this. If we take something and it go into the gut and do all the biological phenomena, it means that it's a natural process. So in nature, how it happens from target identification to exploring the non-druggable target, from a complex system, which is a system biology approach, you got two protein information, and from two protein information, how you go for the inhibitor, that is actually the challenging aspect to design. Here is the another example. This is a very classic example. If you want to work on a system biology based manner, what does it mean? System biology means you have a network of the protein which involved in a particular biological function. So that network, suppose this is your hub, this is your protein which you got to know that if you block this protein, if you knock out this protein, there is a huge effect in the multiple pathways. So you started targeting, okay, this is my target and I will design a small molecule for this target. What happened? Your molecule go and bind there. It associated with multiple pathways. So you can see it binding of this small molecule in this pathway weaken this direction. You normally in biology you say downstream pathway change, upstream pathway change. What does it mean when you bind a particular target in the pathway? It is the meaning actually. So when your molecule bind in the same target, it weaken this pathway, stronger this one, and there is no effect on this pathway. But the different molecule in the same target, when you bind a different molecule in the same target, what you are reflecting, what you are finding, this is now weakened, this became strengthened, and there is no effect. So it means you have to be more cautious when you are playing with PPI interaction, because your small tweaking, your small modeling change induce a huge compact on the rest part downstream pathways. So it is more challenging to understand this process. Another very complex property is associated with this property to deal with the protein-protein interaction. Since protein already have multiple type of conformations, there are side chain dynamics, main chain dynamics, backbone dynamics separately, the folding. There are some structured state, there are some unstructured state. Then intrinsically disordered region. How to deal with these problems? Because protein not always in globular shape. So these are the very complex situation to design a small molecule. So you can see the complexity. When you have a short peptide domain, you can deal with, okay, small peptide, I can understand how many amino acids are there, we can map it. But see, when this peptide from coil to become a helix, when this helix become the whole domain and then whole domain and intrinsically disordered, there is no shape. So when there is no shape, there is no static state, there is no stable state. So how you will design from this interface? It's a very challenging aspect, and we need at least examples to understand this first. So what are the examples are? Possible computational approaches to target interface site for drug discovery. We need examples from nature itself. Since you people, many have heard about they are working in plant science, so this is classic example. Axine, how it works. Axine work in a protein to bind it so that it can provide the facility whether other protein can bind. Rapamycin is one of the classic example. I am giving you more example on rapamycin because I worked on it. So it provides it facilitate these two protein by binding here so that these two protein can bind very nicely. Means if these two bind with the energy, for example 4 kcal per mole, with this small molecule they are binding better. Imagine 8 kcal per mole. So how this happen? How nature planned this thing to design this? Nature also faced this problem, but how they overcome this problem to design some molecule which behaving like a natural glue to contact two proteins. So this is the very classic example. When you treat your molecule with rapamycin, it goes into the cell, it binds its appropriate position, and then it mTOR gets signal, and mTOR bind into the FKBP. FKBP is a protein which is 506 FK binding protein, which is responsible for when organ transplant happen. So in organ transplant, we normally use immunosuppressant, which is normally calcineurin or rapamycin. So these two proteins glued together with the natural way and performing the protein-protein interaction to communicate the message from one protein to another protein. So what is the message from here, from this story? The story is they have a cooperative behavior which provide the high affinity, and cooperativity is the intrinsic property of protein by which it induces the signals. Let's have a next example, which is also a FKBP and rapamycin, but here the partner is calcineurin. So it's a very classic example for showing how these proteins behave. So you can see here, these are written in the area where this rapamycin binds, and this protein is supposed to bind here, but not able to bind because in absence of rapamycin, these two are not able to bind together. But when you treat it with rapamycin, these two proteins bind together. So what rapamycin does? It works as a glue to bind between these two proteins. Additive, you can see. The same story here. When you treat it with these two proteins, FKBP with calcineurin, when you do some organ transplant, Medico treated this. Doctors prescribe to treat with the FK506. Why? Why? Because they want FKBP to bind with calcineurin. And how this happen? This FK506 bind into FKBP, provide a surface so that these can two bind together, and then the cell communication happen. So these are the classic example to bind. By taking these examples, people discovered many small molecule which really works fantastically and which reached into clinical trials. And this molecule ABT737 recently approved by FDA. And this is the first case approved by FDA. The molecule derived from the PPI interface to control the cell signaling process. So what I am doing actually in this area, so it's a very fascinating and challenging area because here you get all this information. So in THSTI, we have a team of mathematical modelers and a system biology team which develop the interactome of your target protein. So here I'm taking an example of AKT protein, which having interaction with lot of many other proteins during the biological function. So if this is your target protein as AKT is the hotspot interface, how you can design a molecule? So just assume this is your inhibitor. When it bind with your target protein, this is not allowed to bind the second partner to this site. Or if you design an allosteric site, it will bind in some other region. It change the shape of your binding pocket so that this partner cannot able to bind it. So by taking these examples, we have three different methods by which we design the small molecule. These are the very fundamental and well established methods, that is virtual screening, fragment based methods, and peptidomimetics. I highlighted peptidomimetics because I'm going to talk about in a more extensive way. about the identification of these hotspots. So in peptidomimetics, first information which is essential to know about is where these two proteins are bind together. What are the zone which is called as hotspot. So hotspot can be your secondary structure, hotspot can be your amino acids, hotspots can be targeted by these two people with these two information. And when you understand these amino acids properties, how they are interacting with other partner, then you can able to design a small peptide which are able to bind with the partner, which not allow to second partner to bind there. So this whole process is called the peptidomimetics. Why this peptide, and what would be the size then? If you drive a structure, if a peptide from the interacting partner, what would be the ideal situation, ideal length of the peptide to bind onto the surface? So from the literature which recently 2016 published, people are very much interested in this area. Lots of recent research are coming. They find that the amino acid set, the peptide set, which belongs to 2 to 10 amino acids in length, they are the most effective one. So we accordingly we plan our strategy. When we have a sequence, this sequence have a straight structure. When we perform the molecular dynamic simulation to get the information about the shape, where this amino acid turn and what kind of shape is possible, then these shape are evaluated by Ramachandran plot, whether phi psi angle are okay or not, whether they are belongs to allowed region or disallowed regions. So all these property we check through a tool with ProCheck, and then we sampled in a different conformation. We performed a clustering analysis, then we performed protein peptide docking, and then molecular dynamic simulation to calculate the delta G. This is another strategy when we don't have any structural information. So these are the Monte Carlo simulation method where your peptide is not able to is able to bind to the target but not stabilize enough. So we have a strategy to point out the residue-wise decomposition, where you have the information which residues are contributing maximum, and those contributing residue we are not going to touch, not going to disturb. So we are focusing on the neighboring residue so that we can staple those peptides to design more stable, more hydrophobic peptides which bind and interrupt the PPI interface. So for that reason, we generated the protocol. We take multiple different snapshots of your peptides. Then we do individual peptide to protein docking, and we calculated all the steps in terms of delta G energy. And when we got the best delta G energy, most stable state of the peptide, then we performed molecular dynamic simulation for each state with different force field. What is the meaning of the different force field here? Means we have to calculate the distance, the angle, the dihedral for all the peptide and protein complexes with different parameters to get the most stable state. Here is the free energy surface where we are getting the most stable state of the peptide, and the most stable, most native state of the peptide we used for the peptide conversion into small molecule, into hit molecule. So these are a very long step process. We are not getting these things in a day. Even people are thinking that okay, running in the simulation it easier stuff. It take lots of high performance computing resources. We have a big nodes, big cluster on that. We have GPU facilities to run this simulation to generate this much statistical data. So overall, the conclusion from the interactome, if you know the main key target proteins which are interacting, you know want to quantify this interaction for the designing of your small molecule. So you first know, this is the summary actually, you first know what kind of secondary structure involved in the affinity of these two proteins. When you get the exact information, which kind of residues are involved there. When you get the information about residues, you convert and mimic those residues into the small molecule which are able to bind, and finally from the peptides, from the helix, you get the small molecule. So this is very interesting and discovering and challenging area to design a small molecule from the peptide. But from the venticlox, since it's approved by FDA, people want to take this challenge to design new molecule which modulate the cell signaling process. Now I'm going to tell you the case studies where I apply these methods, this protein-protein interaction strategy, and I got some recent success. So these are the three different case studies. I successfully patented this also. So these are the first molecule which is autophagy inducers, and here the example is, I know all the information from the chemist people, from the biological people, means both the partner proteins and the peptide information is known. Second example, where I don't have any information about the structure. So to deal with this project, I need to develop my own homology model as well. So here the collaborators only give me the sequences and say, from this sequence you have to generate some small molecule. There is no any crystallography information, there is no structural information available, and no support from the chemist as well. So these are the most challenging area. We get some success in this area as well. And third and very complex is the flexible side. So here these IL-3R, interleukins 3R, we want to develop some agonist which able to bind on interleukins receptor and do the signaling process. So these three projects, which this is our in-house project, which is going in THSTI DDRC. This is working with Dr. Ravindran, which is who is former director of ILS Bhubaneswar, and this is with NCCS Pune. So this autophagy inducers, this work already patented. Bethlehem in the US is a group who is working on autophagy induction. So what autophagy is? Autophagy is a biological process which clear the debris of the cell. And what this Nature paper reported, that if you increase the autophagy process in an infected patient who is suffering with HIV infection severely, and if you treated these patients with these peptides, you can see the clearance viral load decrease significantly. So this is the groundbreaking research, because by inducing autophagy, nobody is reported that with significant viral load. And this Guwahati and et al. In Nature paper, they find that these autophagy induction works very nicely with autoimmune diseases and neurological disorder as well. So it was our interest to understand how this peptide is working to induce the autophagy and reduce the significant viral load of HIV infected patients. So we start from our computational studies to perform the protein docking studies to understand between these two proteins. This is the GAPR protein and this is the Beclin. Beclin is already known as a positive regulator of autophagy in different cases in NEF protein, but with GAPR it was reported first time by Bethlehem. So we try to understand how these two interactions, these two complexes formed, and we quantify all the residues which are involved at the interface, and what are the complexes and which energy they are stable. So from this complex study, by doing all these analysis, we find these top 5 poses which are producing and having significant interaction between these two proteins. Then we perform molecular dynamic simulations to understand, along with the dynamic motion, along with the thermodynamic calculation, either out of these 5 poses, any one pose are stable. So we found that all are stable, but this pose number 5, showing the most stable state. So we took this state for the development of the peptides. We develop our own peptides apart from what Beckley group developed. We generated our own peptide to see whether our peptide, which also derived from the same Beckley protein, are able to control this GAPR, which is a negative regulator of autophagy. And we found, yes, our peptide are also working at the same level. And so we performed this experiment to understand this thermodynamic stability of the protein and peptide. But the intention of R is not to generate one more peptide. Our intention is to convert this peptide into small molecule, because peptide has problem. If it is not bind at extracellular proteins, it will go inside the cell, and the most possibility the proteases will kill this peptide. So your all effort go into vain, because you are not able to deliver your peptide into the real target. So the best solution is, and another drawback is, you have to deliver this much big peptide through IV only. You cannot do oral doses. So we want a molecule which which have a same effect, but you can treat it orally. So what we did, we take the most stable state of the peptide, we try to find out which amino acids are really interacting and have a tendency aromatic residues. We play around all the possible permutation combination. We generated more than 2000 possible compounds, and from 2000 compounds, from calculated energy, we picked 120 novel heterocyclic compounds to perform in vitro cell analysis to see whether these molecules are able to bind here or not. And then from SPR binding, from surface plasmon resonance binding, we find our molecule which derived from the peptide are good enough to bind in 78 nanomolar. So these data are showing that what all exercise we performed, finally we get a molecule which are able to bind on the GAPR and allow the bacillin to let it free so that it can regulate the autophagy process, and by doing so the viral load get it down significantly. So what we did, our molecule bind at the GAPR interface site, and we generated more than 150 hits from the different series. And these three different leads we explored in three different therapeutic area. One is the HIV infection, other one is the cardiomyocyte, which is related to heart, and third one is the NASH, which is non-alcoholic liver steatohepatitis, which is another lethal area, and where autophagy works very nicely. Not to limit our working strategy, we want some repurposing also. So we use this same target to screen our 5.7 million compound library, and we got some hit which are still in vitro analysis. The result not yet done. So these are the result strategy, because I'm keep on saying that the peptides which either published paper, Bethlehem group published, or our peptide are able to clear the viral load. But is it possible that it also bind, apart from autophagy induction, it doing something else? So when this idea came into the picture, maybe it bind with the other protein of the HIV, maybe RNA integrase, RNA protease, reverse transcriptase, or any other protein. So we did tested our compounds with different targeted protein, which basically known especially against integrase, proteases, and reverse transcriptases. And we find that our molecule not only inducing the autophagy, it also bind with the integrase and do the inhibition process. And that actually the cumulative effect of this small molecule, because it able to induce the induction and it also control the integrase. So it's dual activity by which we are getting the significant viral load down. So why our integrase molecule is behaving differently, means how they are binding and how it is different with other integrase reported inhibitors. So they are differently binding. If you see, these are the raltegravir, which is very well documented series of the inhibitors against integrase. But our molecule, you can see DR1, which bind in a different direction. And you see, this direction is important because it's not only interacting with the different set of residue, but having more water-mediated contacts. So it is more transient and stable state of the complex, which provide the additional stability and not going to this direction, reported inhibitor direction. It attain the same binding pocket but in different orientation. So what is the benefit of this? The benefit of this, our molecule also effective on resistant mutant also. So not only it blocking the wild type, it blocking the mutant strain as well. So we get the one shot, we get the three benefits. First, it induce the autophagy, it inhibit the integrase, and it is still active on the mutant strain also. So all these three positive, we submitted this into BIRAC, and we get funded from the BIRAC to develop these molecule for the further development, to consider as a lead candidate for drug designing, for drug development. And this is the current status of the molecule. It is in the second stage of BIRAC, so it's well funded. And these are another classic example. We performed in vitro studies. We generated POC. POC means we tested our molecule in rat and mice, and we found there is a significant enhancement in heart muscles when you treated with these molecules. And for NASH also, which is a liver cirrhosis, before cirrhosis, we used through treatment, and we see a significant reduction in triglycerides and lipid content, and the liver perform functionally better when it's supposed to be not infected with the disease state. So with this molecule, we have the significant 50 nanomolar status of this molecule, which reduce the lipid content significantly. These two works are already going on in THSTI. And these are the overall conclusion from first example of PPI interaction. So we, from this interface, from this protein, from hardcore computational studies, we found a molecule which is active against HCV able to reduce the viral load, which is active against cardiomyocyte also, and which is active against NAS and other area like neurological disorder and others we are now exploring. So the take-home message is from the PPI interface, from the computational study, you can design your research area which are able to modulate and translate not only for the discovery point of view, but you can develop your small molecule for the further advanced stage of your drug discovery. This is the second example where I'm showing you an example where nothing is reported. Means I don't know any partner. So no crystal structure of partner protein A, no crystal structure of protein B, no NMR data, nothing. So from the sequences, when this project came to my hand, that this protein, this case related to sepsis. I don't know how many of you is aware with the sepsis, but this is a very common problem, especially in hospitals when people get died because of multiple organ failure. And sepsis is related with the inflammation. When the inflammation level is too severe, your organ cannot able to control, and then the only result is the death. So sepsis is a very critical stage, and ICU is one of the responsible area where sepsis normally happen. So to control this inflammation, what patient actually need? It need nitric oxide. And in our cell, three nitric oxide producers are INOS, ENOS, and NNOS. But here the case is the INOS, which is really responsible at the case of organ failure. And the solution of this critical complexity is to search more nitric oxide donor. But nitric oxide donor is, if you are rich enough, you get it, but their T half is very less. T half very less means they are not used for a longer time. So even though you are able to get the nitric oxide donor, but you cannot get it proper solution, and the result is the same, the death. So this project need a kind of some endogenous strategy to develop to generate more nitric oxide endogenously. What does it mean? It means your body are able to produce, when you have more inflammation, your body itself can generate more nitric oxide. So Dr. Ravindran and his team find out a protein which is called NOSIP. NOSIP is a negative regulator which is associated with the mortality in sepsis, and this NOSIP protein bind with INOS to block nitric oxide production. So when he came to the collaborative project, he told me to design something from the protein-protein interaction interface so that NOSIP is not able to bind with INOS. INOS get it free at the infectious stage so that it can generate endogenously more nitric oxide. So what is required? It required to keep this NOSIP away from INOS. And this is only possible when you know what area of NOSIP interacting with INOS. So if you know the area of NOSIP interacting with INOS, you can dig out some peptides from NOSIP which you can throw to INOS, and this INOS peptide will vice versa. When you know the area from INOS to NOSIP, you design peptide from INOS, throw into the NOSIP, and this way NOSIP INOS peptide binding allow this INOS to get it free. So we started this work by doing by first establishing the structure, because the structure was not known. So we have to generate the structure, 3D structure, to understand, to perform the protein-protein docking. And so, this is the critical case where we don't have any experimental background. So I used 7 different algorithms. These 7 different algorithms are used only just because to increase the robustness of the result, to increase the affinity and accuracy of the result. And what we got, we generated the NOSIP and INOS structure, and then we performed protein-protein docking, and then we extracted the most stable interacting complexes of INOS and NOSIP. And from there, I extracted the INOS peptides. Why INOS peptides, not NOSIP? The reason is I want to block NOSIP, not INOS. So if I have to dig out the peptides from INOS, it will go and bind with NOSIP. So NOSIP can get their complex state, and this way INOS get free. So we identified these 4 peptides from both the sides, and then we performed the experimental state, whether these peptides are able to bind or not. So from SPR analysis, they are showing they are bind very nicely in nanomolar range. Another strategy, when this green one is the NOSIP and the red one is the INOS, and this red one is the NOSIP here. So when NOSIP bind with the INOS, without binding here, if you see this highlighted zone, this state is in disordered part. There is no secondary structure. You see in the blue. When INOS came into the picture, when it bind with the NOSIP, this blue is now here. You see, without partner, it is in disordered state, no shape, no existence. But when the partner come into the picture, they start taking shape, they bind and support into the binding. So from these information, what I find a disorder to order transition. Disorder means no structure, ordered means structured. So this no state, loop state to secondary structure helix formed. And this zone, if I am able somehow to block this transition, I can able to block this interaction between these two. So from both the strategy, what are the inhibition strategy now? To locking the transition from disorder to helix through small molecule or peptides, so that this transition cannot convert. And since this transition not happen, INOS not able to get the partner interacting area and it will not bind. Second is to design some peptide to kick out this INOS from NOSIP interface. So these two strategy, when I came to the picture, we started doing screening to get some molecule, and this is a huge library. So we came into the picture from by screening in each and every step. So from 5.7 to 25,000, 3,000 something, then 250, and finally the 50 compounds on the basis of different cutoff parameters to perform MMGBSA analysis. What is MMGBSA? Is the explicit solvent molecular dynamic simulation, where you can get the energy when these two complexes are formed. What is the energy ratio? And then with water, what is the energy ratio? With water is important because your system rely on the water system and the concentration. So from 50, we filtered out then 18, and from 18 we performed the biological experiments on 12 compounds. The in vitro is already 7 compounds are in working perfectly. These 7 compounds now are tested in vitro in animal models. These are the compounds which are working. You can see the KD values, they are all in nanomolar range. And this is the third and last story. This work with Professor Mohan Vani in NCCS Pune. He's working on osteoporosis. So for osteoporosis, the mechanism is osteoblast and osteoclast. These are two types of the proteins which actually always remain in the homeostatic condition. If there is any disturbance in the blast and clast differentiation, it means you are going to face a problem related with rheumatoid arthritis, bone depositions, calcification problem, porosis, lots of problem related with calcium. So he is working on this arthritis problem long back. He's more than 10 years, and also it is important for a veterinary point of view, because for horse is important. So he wanted to generate a IL-3R agonist which are able to bind on interleukins receptor, so that interleukin can bind there and generate a signal by which more blast differentiation can occur. And this class blast differentiation, we need to block a molecule which able to bind here and can generate this time t-half in a more proper way. So since there is no any more detailed information are available, we not only to achieve this IL-3R agonist designing, we studied in more extensive way what all interleukin structures are available, IL-6, IL-4, IL-13, IL-17, more detailed analysis, because some of them are homologs and they are matched to each other. And finally, we got the exact partner where IL-3 bind with IL-3R. And what are the reasons? You can see here the two helix which are bind very nicely with these loops actually. And if we have somehow designed something which are able to bind here as a additive, so we want to design a glue so that these molecule, this IL-3 can bind more efficiently and with increased t-half value. So we designed it, this molecule. It is more challenging because these loops are very flexible in nature, but we somehow manage, and we find this peptide which is working very perfectly fine with these highlighted residues which are contributing maximum. And at the stage to whether this blast class differentiation is working perfectly or not, we have to monitor an experiment where we see the p-stat monocytes derived PBMC's phosphorylation status. If a peptide this bind on this IL-3 bind are able to bind on IL-3R surface, the phosphorylation status must be high. And from experiment, we find that our peptide work perfectly fine as for phosphorylation status with the peptides significantly high. So we achieved our one of the target, and these are the conclusion of my work, that we designed some molecule which are able to bind, control the NOSIP to control the sepsis progress work. From small molecule virtual screening, we designed some molecule which are work as INOS-NOSIP stabilizer. I'm designing some NOSIP blocker inhibitor, but accidentally from the same library, I also find some molecule which are able to strain this interaction pattern. So this was the opposite result. It's a serendipity. And when I discussed with Dr. Ravindran, he said, okay, this is not a bad example. We can use it as a drug candidate for this disease, chronic OPD pulmonary disease, COPD. So this, by accident, it's open another venue where this molecule can work on. So these work are in the process, and it reached up to clinical trial. Dr. Ravindran is planning to send it because it's working perfectly fine with the rat model. And now IL-3R agonist is in intermediate stage, and I'm planning to generate more these two work, glue effect and seizures effect, for the futures. These work are in the process. Overall, this is my working area, my blueprint. In THSTI, if we got to know the information about target protein, we have different PPIs working on this area to identify the target on the respective gene. And if we also have the information about ligand, well and good, most welcome. If we know, we have to generate this information and these are the tools. For the molecular dynamics, we have CPU cluster, GPU cards, multiple tools. For bioinformatics, we have these in-house scripts we wrote down to generate our own pipeline, which is basically written in Shell script and Perl. Then computational chemistry, I have Schrodinger, so we can do all the QMMM analysis, QSAR, computational screening analysis to know all the electronic structure, how these changes happen. And these are the resources. I have fragment-based library, FDA-approved library, and if something happened to some click at atomic level, we have a medicinal chemistry and experimental biologist team to directly achieve up to lead. If not, we have to reverse this process to design to study in more detailed atomic level these properties of molecule to design more potential and targeted inhibitor. So that's it, and these are the collaborators. At the end, I want to acknowledge all the people who really contributed me. These are the works apart from what I have presented today. Dr. Ravindran with INOS-NOSIP, IL-3 Mohanwani. But I'm also working on kinase inhibitors, some plant products also, gibberellins with IRI, and with chemistry on dengue and DNMT and sirtuins with Dr. Bhoja. I'm really thankful for my team members, Dr. Dinesh, Shilpa, Samina, and some projects I'm working on HIV with Dr. Milan and MTV with Ramandeep and Krishna Mohan. This work I'm working with and PD1 work with Dr. Avasti and with Dr. Joshadev here in IASST. I'm really thankful to him for joining hand on this project, and Dr. Sankar on dengue project. These are my students, and who really worked a lot in this aspect. And overall the funding, thank you, and thank you all for your attentive listening. Thank you.